Right. Good morning, everyone. I'm Paul Saboch from the Blood and Marrow Transplant Division. For residents, I wish we meet more often, and feel free to request electives with us. But this is not a pitch for electives, but rather a grand rounds. And it's my absolute pleasure and honor to introduce our speaker today, Leslie Kean from Boston Children's and Harvard University. Very briefly, she has had training at Case Western, Cambridge, Stanford, and eventually Emory University, where I think she got her most formative training, where MD PhD was followed by postdoc years with one of the giants of organ transplant biology, Chris Larsen. She built on her biochemistry work in PhD and gradually has become one of the leading experimental transplant biologists, utilizing some of the rhesus macaque animal models that have been so instrumental for solid organ transplant. Just the key words of her work in terms of what you might expect today. So immunology, system biology, transplant tolerance, and graft versus host disease, I think, are the key words that I would use. And really, I think Leslie represents maybe the most productive and impactful transplant biologist of our time. And she will really talk about us how concepts evolve from the bench and then go through various animal models and eventually take us to successful FDA IND approvals. So just last, her career has taken her from Emory briefly through Seattle at the Fred Hutch Cancer Center. And since 2018, she's now at Boston Children's, the director of the Pediatric Stem Cell Transplant Program. She has multiple leadership roles. She's chairing the Pediatric Transplant and Cell Therapy Consortium, but she's also really internationally leading various scientific programs and efforts. So without further ado, I would rather have her take over and talk about the road she journeyed. Her title is going to be From Bench to Bedside to the FDA, Cell Costimulation Blockade for the Prevention of Acute Graft versus Host Disease. Thank you, Leslie, for making the extra effort. COVID has been hard on everyone, and just really appreciating your presence. Thank you.
Great. Thank you so much, Paul. Can you all hear me? Yes, great. And hello, audience in Pittsburgh. I'm so sorry that I'm not there in person. I have been exposed to COVID and am symptomatic, and really felt like I just didn't want to share the wealth of COVID with all of you. And I know it's again, we're in a new era of COVID surge. So I hope you all are healthy too. And I really appreciate the invitation here. I am a pediatrician, as you know, and now I'm also the mother of a pediatric intern. So the generations continue in my family of pediatrics. And I think one of the through lines that I'll talk about today is sort of bringing a pediatric eye to what we do in translational and clinical research. So I titled my talk From Bench to Bedside to the FDA, T-Cell Costimulation Blockade for the Prevention of Acute GVHD, because over the last 20 years, it's one of the things that my lab has really focused on. And I think it's a good example of how translational work can actually get to the patient. And so I want to talk to you about that story, and also how our role as pediatricians is so important in bringing children along on this road, which is often much harder to do for children than it is for adults. Here are my disclosures. And let's start with a case. So this was actually the young man who really changed my research world. As Paul said, I was working in the area of transplant tolerance and studying solid organ transplant tolerance under Chris Larsen, who was a giant in the field, but had started being a pediatric transplanter. And this case really was the inspiration for me to build a non-human primate model of GVHD. So this was a 16-year-old young man that I took care of my first year as attending. He was in 10th grade, football player, just an amazing young man. He was totally well, star of his football team until he was diagnosed, as often happens in teenage years, with acute myeloid leukemia and required a hematopoietic stem cell transplant in his first remission. He was an only child, so he had no siblings that were possible to be matched for him, and he was of an ethnic minority. And I'll show you in a few slides, but as we often see, patients from ethnic minorities have a lot more diversity in their HLA genomes and often have no HLA matched donors in the registry. He did have several 7 out of 8 HLA matched donors. So, HLA matching is done on a variety of scales, but one of the most common is using 8 alleles. So the 2 alleles of HLA A, B, C, which are class 1, and the DR allele of class 2. And so he had 1 mismatch, but what we know is that patients with even 1 HLA mismatch historically have been at high risk of death after transplant. He did undergo the transplantation with his 7 out of 8 donor. He had high intensity conditioning with standard conditioning regimens of busulfan and cyclophosphamide. He received what at the time was standard, which was a calcineurin inhibitor, tacrolimus, and methotrexate alone for graft versus host disease prophylaxis. He engrafted with his donor cells on day 21, so that was a successful part of the transplant. However, on day 35, he developed a rash over his entire body. A biopsy was done, which confirmed skin GVHD, and he was started at the standard for skin GVHD at the time, which was 1 milligram per kilo of methylprednisolone steroids. However, diarrhea developed at day 40. His steroids, which is really still this was almost 20 years ago, steroids are still the first-line agent for our patients, increased the dose to 2 mg per kg. However, this young man, despite increased steroids, continued to have high volume bloody diarrhea. Endoscopy was performed. It confirmed acute GVHD. And unfortunately, he developed what we call steroid refractory GI GVHD, for which morbidity and mortality are very high. His skin disease did respond to the steroids, but as often happens, the GI disease didn't. He was treated with everything we had. We threw the book at him, 5 second-line agents, but none resulted in a response. He had to stop eating, of course, because GI GVHD really creates a situation where you can't absorb nutrients. He was maintained on TPN and escalating immunosuppression through day 170. But as often happens in our patients that are being treated with high intensity immunosuppression for GVHD, he developed sepsis, was found to have both adenoviremia and fungemia, transferred to the ICU, and then 20 days later, this sweet young man died in the ICU of multi-organ failure. And what really is so painful for his family and for us as his treating team was that, we did our job in terms of keeping his AML in remission. So his AML was in remission for the entire post-transplant period, but it was a complication of this cure that killed our patient. And seeing this patient and doing non-human primate work really changed my whole sort of career. And I decided I needed to focus my career on solving this terrible issue of graft versus host disease. And I just gave you a vignette of one patient, but the CIBMTR, which is our registry for bone marrow transplant, keeps exquisitely detailed data on all our patients. And you can see that for patients like him with AML, the probability of survival after transplant is still really insufficiently high, with even early phase disease having only about a 50% long-term survival after transplant. In the field of bone marrow transplant, we actually know exactly why people die after transplant, because the CIBMTR keeps very detailed records. And here is a pie chart showing why patients die after transplant for unrelated donor transplant. And you can see that GVHD, which I'll be talking about, has been one of the largest causes of non-relapse mortality after transplant. But I do want to direct everybody to this large blue piece of the pie, which is primary disease, which really means relapse of their primary disease. And you can see that it remains the most important cause of death after transplant. And so for people like me who really focus on GVHD, if we focus on GVHD and ignore primary disease, I would say that we're really not doing our job. And we have to learn how to control primary GVHD without negatively impacting protective immunity, which can impact both primary disease and what this young man finally succumbed to, which is complications of infection. And so when I sort of focus the audience on GVHD, I often draw a circle like I have here that overlaps with infection and primary disease, because the tools that we have, and they have been really blunt tools until very recently for controlling GVHD, are broad immunosuppressive agents that often will have untoward effects on protective immunity, both for infections and for disease relapse. So let's take you back to the time where this patient was transplanted. And this is from a paper from around that time, which really nicely cataloged the risk of mortality after transplants of different HLA disparities. And I've boxed in red here the transplant-related mortality from that era. If you had an 8 out of 8 transplant, so that's a fully HLA matched transplant, was still fairly high at 36%, but you can see the transplant-related mortality for a 7 out of 8 was as high as 45%, and that's what we saw in the patient I just talked about, with severe acute GVHD, that's grade 3 to 4 GVHD, occurring in almost 40% of these patients. Really striking data to suggest that we had to do better. But what about these patients that only have a 7 out of 8 matched donor, that don't have that lower risk 8 out of 8 matched donor? Well, a study that was published in the New England Journal several years ago now looked across the world at different ethnicities to see what was the degree of matching that one could find in the donor registry for people of different ethnicities. And you can see that if you're of a white European background, at that time, your chance of finding that fully matched donor in the registry was quite high. But for essentially every single other ethnicity, the chance of finding a HLA matched donor was below 50%, and really quite dismal for African-American patients because of the genetic diversity of their HLA. Very difficult to find a 8 out of 8 donor. But if we even allowed for one mismatch, that 7 out of 8 donor, and that's what we found in our patient that I talked about, really almost everybody is going to be able to find a donor in the registry. The problem being that historically, these have been very difficult transplants to get people through successful because of complications such as acute and chronic graft versus host disease that cause high rates of transplant-related mortality. So let's dig a little bit into graft versus host disease. It's the complication of transplant that I'm going to be talking about today. So we know a lot about graft versus host disease, but there's still a lot that we don't know. What we know, and I'm going to focus on acute graft versus host disease here, is that it has been one of the largest causes of non-relapse mortality after bone marrow transplant. It remains a clinical diagnosis. So really it's about what your volume of diarrhea is, what your bilirubin is, what your degree of rash is. However, there are several serum biomarkers that are now helping us with the diagnosis, but we still only have partially effective prevention and treatment. The 3 canonical organs that define graft versus host disease in its acute form are skin, where you see a rash, liver, where you see infiltration, often into the portal triad, which gives you a cholestasis, often identifying as increases in liver enzymes and bilirubin, here showing jaundice, and then GI GVHD, which is the biggest cause of mortality of acute GVHD, which causes destruction of the GI tract. What we don't know really dwarfs what we know. We still don't know what drives the organ-specific manifestations of this complication, and we don't know how this differs from what we can see in the blood, the easiest tissue to probe. We don't know still how to differentiate the T cell expansion that has to occur after transplant, T cell reconstitution from T cell pathogenesis. And finally, and what my lab is really focused on, is how we identify the best targets for therapeutic intervention. So as I said, when I was treating this patient and doing a lot of non-human primate work for solid organ transplant, I decided to shift my focus of my lab and develop an acute GVHD model. We first published this model in 2010, and it's become a real workhorse in the field for understanding mechanism and testing new therapeutics. So I'll just go over a little bit about the model because it really led to the use of co-stimulation blockade in GVHD prophylaxis that I'll spend the majority of this talk discussing. So the non-human primate acute GVHD model was designed to fully recapitulate what we do for patients in an experimental model. So we have recipients and we have their donors. These are family members for which we can do detailed MHC typing. Usually what we use in this model is a, sorry, I'm gonna, one second. I'm just gonna hide the video. Oh, sorry. Okay. Use haploidentical donor and recipient pairs. We have a clinical grade apheresis machine that we use to have a PBSC transplant. We transplant after myeloablative conditioning using TBI, using chemotherapy, just as we do in the clinic. And then we give both standard of care and investigational GVHD prophylaxis. We do really detailed supportive care for our patients, including antibiotics, antiviral medications. Our recipients are on TPN, really quite similar to what we do in the clinic. And then what really is a value add over what we can do in the clinic is we can do immunologic monitoring on these patients to really understand the mechanisms by which their GVHD occurs or can be prevented or treatment with novel and standard therapies. So the added value of this model is that it can support immunologic studies in a very evolutionary proximal species in an experimental setting that most resembles clinical transplant, but enables high resolution multi-tissue immunologic monitoring to really get at that mechanism so that we can design new therapeutic strategies. So here's what we see when we use the non-human primate model. Again, as I said, for humans where we look at skin, GI, and liver, you can see that in a setting where we haven't immunoprophylaxed the animal, we see very severe early GVHD that very much resembles acute GVHD in human. We see rash that comes up very early. Again, this is a haploidentical transplant without immunosuppression here, so we can see the natural history of the disease. We see diarrhea and GI findings similar to acute GVHD. We really just start to see a hint of liver GVHD clinically, but when we look pathologically, what we can see is in the GVHD animals, as opposed to two different controls, healthy controls and autologous controls, we can see that we see pathologic findings in the skin and in the liver. It just hasn't quite ticked over to clinical liver GVHD, and then in various parts of the GI tract. This GVHD happens with the acquisition of almost full donor chimerism. So it's the donor cells that are causing this. And this leads to quite severe disease that necessitates euthanasia within about 10 days of transplant, compared to autologous controls that actually go on really forever. I just show you out to day 60 here, but they can go on to years. So really a nice recapitulation of human GVHD. Now, we've done a ton of immunology here, but I just want to show you some canonical signs of GVHD in the non-human primate that really mimic what we see in our patients. So first of all, we see a shift towards antigen experience phenotype. So the T cells in these animals, which is what the T cells cause GVHD, really shift from naive T cells to more central memory, effector memory T cells. Those naive cells become activated. We see a highly proliferative, but also pro-apoptotic phenotype. So Ki67, a canonical marker of proliferation, is almost fully upregulated on these cells, and they downregulate BCL2, an anti-apoptotic marker. We see excessive cytotoxicity with high expression of granzyme B and perforin. Here I'm showing you granzyme B upregulation in recipients with GVHD. And then, as we often see in patients, we see a degradation of the T regulatory to T conventional ratio, suggesting an imbalance in our pro-inflammatory to suppressive T cell milieu. What the value added is of the non-human primate model is we not only can look in blood, but as I said, we can look in all of the different tissues in the animal. Here's just a survey of multiple different tissues showing that all of them have T cells that are undergoing high degrees of proliferation as measured by Ki-67 and high degrees of cytotoxicity as measured by granzyme. And here we can do immunofluorescence as well. And we're increasing our sort of capabilities of doing spatial proteomics and transcriptomics in these experimental animals. So what we found when we first started doing these studies in non-human primates was that you had a very activated T cell milieu, just like we saw in mice, just like we saw in patients. And indeed, when you think about what are the potential mediators of this highly sort of Th1 activated T cell, what you see actually from the literature, and a lot of this did come from solid organ transplantation, is that what we know about T cell biology is that there are multiple T cell co-signaling pathways that could potentiate this T cell activation, and therefore are actually potential GVHD targets. And I just include sort of a smattering of these co-stimulation pathways here. And as I said, in solid organ transplant and autoimmunity, people have been looking at these for many years, and indeed in GVHD too, but not in the non-human primate model. And so in my lab, just some examples of different targeted pathways that we've looked at in the non-human primate that are really quite interesting in terms of targeted ways to control T cell activation. I'm going to spend a lot of time on the CD28, CD86 pathway, but we've also shown that by targeting 4-1BB or CD137, OX40 ligand, CD40 ligand, these all actually are very important in T cell activation causing GVHD. And we've got a lot of work to do in the future. But I want to tell you the specific story of the CD28, CD80, and 86, since that's recently been FDA approved. So when we started this work, what we knew was that there was a agent called abatacept that blocked the CD28, CD80, 86 pathway that had been approved for rheumatoid arthritis. And so this was an agent that was gaining experience in the field of autoimmunity with a really good safety and efficacy history. And blocking CD28 and CD86 is really important when you're thinking about T cell biology, because this pathway, while there are multiple pathways that I just showed you, this pathway is one of the most critical for T cell co-stimulation. So what do I mean by T cell co-stimulation? Well, when we think about T cells getting activated, here's a cartoon of a T cell here and an APC here and a scanning micrograph of the same interaction here with there's the naive T cell and the antigen presenting cell. You can see that signal 1, something that we call signal 1, is really the major signal by which you get an antigen-specific activation, where the MHC linked to a peptide on the antigen-presenting cell is seen by the T cell receptor on the activated T cell. But what we know is that if signal 1 happens without other signals, you fail to get T cell activation. Signal 2 is what we call the co-stimulation signal. As I just showed you, there are actually multiple co-stimulation signals, but one of the major ones is through CD28 on the T cell and CD80 and 86 on the APC. And then what we have is signal 3 is really cytokine translation and secretion, which also leads to T cell activation. But what people found was that by blocking CD28, CD80, and 86 signaling through abatacept or CTLA-4 Ig, you could block signal 2. And even if signal 1 and signal 3 were intact, what you found, at least in experimental models, was inhibition of T cell proliferation and effector functions. And in the setting of autoimmunity, what people found was that it could be very effective at treating disease. And indeed, the field of GVHD had started looking at CTLA-4 Ig or abatacept many years before. Bruce Blazer's lab, with who I collaborate closely on many of my studies, long before I was doing this work, showed that CTLA-4 Ig in a mouse GVHD model could really improve outcomes in terms of control of GVHD. It was hard for the field to move that to the clinic, though, because there was a lot of concern over how this agent would act in the setting of transplantation. But when we showed in a non-human primate model that, again, is so close to clinical translation, that CTLA-4 Ig containing regimen could, even in a non-human primate model, improve GVHD. And this we published in 2010 and is shown here in this survival plot. We were able to get the interest of the company to at least give us the drug to start exploring this in patients. We did a pilot study of 10 patients that looked really good, and this really launched our clinical studies that I'll focus the rest of my talk on. So after the pilot study, we designed a study of abatacept really to take this primate work and propel it to patients in need of unrelated donor transplant. ABBA-2 was built on the pilot study ABBA-1. I was the IND sponsor of this. This was funded in large part by FDA R01 through the NIH. We did get funding later in the study from Bristol Myers Squibb, who provided the drug for the study. The primary objective of our trial was to determine the impact of abatacept on the incidence of that early severe acute GVHD like our patient had. This is grade 3 to 4 GVHD before day 100 when added to that standard prophylaxis regimen of TAC methotrexate. And we wanted to target both 7 out of 8 transplants of very high need transplants and the more common unrelated match unrelated donor, or 8 out of 8 transplants. We published this work in the JCO in 2021. So this was our study design for ABBA-2. One of the things that I really want to bring forth here is the real need to do randomized studies in all diseases, but especially in transplant, where a lot of our toxicities really rely still on clinical diagnosis. So we worked hard to do a randomized double-blind study of our 8 out of 8 HLA match patients, where half of them got the abatacept intervention. In this trial, we added it to standard prophylaxis, and we gave only 4 doses of abatacept at the FDA-approved dose for rheumatoid arthritis, 10 mg per kg on day -1, 5, 14, and 28. Compared in a blinded fashion to patients that did not get abatacept but got placebo. We would have loved to do a randomized study of the 7 out of 8 itself, but it was actually deemed to be ethically unacceptable because those patients, just like the patient I talked to you about at the beginning of the talk, were so highly likely to die of GVHD that it was really determined that we could not randomize them given our encouraging results of our pilot study. So instead of a randomization here, we compared them in a pre-specified manner to that registry I talked to you about, the CIBMTR registry, and I'll show you that data in the coming slides. So the study design for ABBA-2 was very standard for a bone marrow transplant study, except for most clinical trials, as you know, don't include pediatrics. But as a pediatric team, we worked hard to allow children to be included on the study. And I think it was really important in terms of the FDA approval. So in this study, we included children aged 6 and older. We looked at patients that had hematologic malignancies. And again, they either had a 7 out of 8 or an 8 out of 8 donor. We had 4 different conditioning regimens. So these really encompass the standard conditioning regimens that we use for both adults and pediatrics, with mostly myeloablative, but we did have one non-myeloablative option for patients that really could not tolerate a fully myeloablative transplant. So first, let's look at the demographics of the study, which I think are really important. So first of all, remember, I'm going to show you the 7 out of 8 demographics first. We had a good mix of males and females, but what was really striking and really is in agreement with that New England Journal article is that we enrolled fully 39% of ethnic minorities on this study. And we also, of course, enrolled our 8 out of 8 cohort. And you can see the difference between enrolling 7 out of 8s and 8 out of 8s in a trial. And the 8 out of 8 cohort just was predicted from the New England Journal article, only 14% ethnic minorities were eligible to find 8 out of 8, could find 8 out of 8 donors, but 39% of patients could of ethnic minorities were on the 7 out of 8, really stressing how important it is to do these mismatch unrelated donors so that we can include all of our patients on our trials. And then also we were able to enroll 30% pediatrics on this trial. This is huge because there are so many more transplants done for adults versus peds. The way we did this is to sort of skew our participating centers. So we had many more pediatric participating centers than adults. So even though we transplant fewer patients on average, we were able to enroll 30% of our patients in the pediatric age range, which really made a difference in terms of the FDA approval. And I'll show you that in future slides. So let's talk about efficacy. What were the key efficacy results of this trial? So I told you about grade 2 to 4 GVHD and grade 3 to 4 GVHD, which is the severe GVHD. So if you took moderate to severe GVHD, for all of the plots that I'll show you in the next few slides, blue is the control. So this is the CIBMTR controls that did not get abatacept, and red are the abatacept arms. And you can see that there was and right now I'll show you the 7 out of 8 cohort. And you can see that for even moderate to severe GVHD, there was a significant decline in the rates of acute GVHD in patients getting abatacept. Now, this was interesting, but really not super striking. But what was so striking about the 7 out of 8 cohort is by giving just those 4 doses of abatacept, we essentially eliminated that severe, highly deadly GVHD in that 7 out of 8 cohort, that GVHD that killed our patient that I talked about at the beginning of the talk. And indeed, because of that really promising impact on severe acute GVHD, at 100 days, or this is actually out to 180 days, our acute GVHD, severe acute GVHD survival for the patients receiving ABA was fully 100%, really strikingly improved compared to 57% for the standard patients. Now, while the 4 doses of abatacept really did a great job for acute GVHD, we actually didn't impact chronic GVHD at all. And we did very detailed PK monitoring on the ABA2 study. And this is actually, and what this showed was that potentially the reason that we didn't impact chronic GVHD is that patients didn't actually have any more ABA on board at the time that chronic GVHD, which happens around day 100, starts. So this is our PK monitoring, which shows that we can get really good levels of abatacept up to day about, let's say, 40 with the 4-dose regimen, but after that, it really waned. Several studies since the ABA2 have shown that for non-malignant disease, that if you extend ABA out, you really can control chronic GVHD as well. Given the fact that we really believe that randomized trials are very critical here, we're now running a trial that's actually halfway through its enrollment, so enrolling very well, that is another randomized double-blind study, this time comparing 4 doses, that's the ABA2 dosing regimen, to 8 doses of abatacept to see if we can actually conquer chronic GVHD as well with this regimen. But despite the fact that we did not impact chronic GVHD, this really impressive control of severe acute GVHD in the 7 out of 8 setting was enough to really significantly improve our survival outcomes here. So this is out to 2 years post-transplant. You can see that the non-relapse mortality in the ABA arm was significantly improved compared to controls, relapse-free survival, and I'll show you the relapse rates, which were also really promising, was much better in the ABA versus control, and so was overall survival. This was a real win, and actually really better than we could have even hoped for the 7 out of 8 patients. Now, 8 out of 8 patients have less risk of GVHD, but they still get this, and abatacept helped those patients as well. So acute GVHD, grade 2 to 4, was significantly improved. Grade 3 to 4 GVHD was halved with abatacept. Our trial was actually a phase 2 trial, so it was actually powered to a P of 0.2, so it met the trial endpoint for grade 3 to 4 GVHD. You can see the rates are quite low here. And severe acute GVHD-free survival is also statistically significantly improved with abatacept, and really quite important for our patient that steroid refractory acute GVHD in the 8 out of 8 setting was also significantly improved with abatacept. So this combined 7 out of 8 single arm study with the 8 out of 8 really rigorous randomized double-blind control study really convinced the FDA that this was a good therapy. We received breakthrough therapy designation in 2019. We got priority review in 2021, and abatacept was approved for GVHD prevention, the first GVHD prevention agent approved in the history of the field in December of 2021. And what's important here, as we think as pediatrics, the FDA actually approved ABA for GVHD prevention first in the field down to age 2. That wouldn't have happened if we had not enrolled the pediatric patients on this study. And it really, I think, underscores as pediatricians how we have to advocate for our patients, because they're often left out of these trials. But if we do advocate for them, we can get approvals even for pediatric patients. Now, I've shown you the efficacy indicators, which are so important for getting approval. But really, I think what led to was a big part of the FDA approval was that the safety indicators were also very promising for abatacept. So after transplant, you have to think about engraftment. We think about neutrophil engraftment and platelet engraftment. We didn't have that data for the CIBMTR, so I'm just going to show you the ABA data for the 7 out of 8, but I'll show you both the ABA and the placebo for the 8 out of 8. You can see that neutrophil engraftment was very robust in the ABA patients. And indeed, when we look past neutrophil, and as was platelet engraftment, I didn't show that here, but when we look past neutrophil engraftment to all immune cell engraftment, we actually see really good engraftment with abatacept. This is quite different than with some other GVHD prophylaxis regimens, and I think led to a very robust safety signature for this drug. So if we look at total white cells, neutrophils, which I already showed you, lymphocytes, remember, this is a T cell directed agent, so we're very interested in what it might do to lymphocyte engraftment. No change, B cells, NK cells. And then if you hone down to the T cells, and we'll look carefully at this as we get to the why of why abatacept worked in this study, you can see that the T cell, total T cell, CD4 and CD8 engraftment were all very robust with abatacept, which likely led to a very good safety profile. One of the key things that we looked at in safety in terms of infectious disease, remember that pie I showed you before, we've got to be able to control GVHD, but not have a big risk of infection or relapse. One of the big infections we worry about is CMV. The 7 out of 8 patients had very similar CMV as what you would expect. And then the 8 out of 8, where we had this nice control arm, we could see no significant difference in CMV reactivation or disease in patients that received abatacept. EBV reactivation is another potential infectious problem in our patients. And again, no significant difference there. I think one of the things that was most striking and really led to the survival advantage of abatacept was that it had a very good safety profile for relapse. So this is 1 year. I'm going to show you 5-year data in a second, where you can see that red is abatacept here, black is control. That ABA, when you add an immunosuppressive agent, you really worry that you may improve GVHD, but at the cost of relapse. And a lot of agents have shown that. So we were very concerned about that. Obviously, it's one of the reasons that we wanted to do a controlled trial. But what we could see that compared to the registry, we did not see any increase in relapse in ABA. In fact, even a hint of a lower rate of relapse. Again, this is against registry data, so you have to take that with a grain of salt. But again, no increase in relapse in the ABA patient. This is out to 12 months. But you can see here, this is our 5-year data, so 2,000 days post-transplant, and you can see that the rates of relapse from the ABA2 patients are still really good. So 8 out of 8 placebo versus 8 out of 8 ABA, you see no increase in the relapse. And what we've seen, and this is now consistent, is that the 7 out of 8 patients seem to have some protection when they receive ABA. And this is potentially because they have more alloreactivity that can cause graft versus leukemia. We're still looking into the biology here, but this is an effect of abatacept that has been stable and is very encouraging in terms of the safety of this drug. So one of the things that I think is also critical when you're doing novel clinical trials is to have mechanistic studies that are baked into the trial so that you can understand the why of either success or failure as you do these studies on our patients. And so for the abatacept clinical trial, we had a very detailed mechanistic studies pipeline. We enrolled 185 patients, of which 116 received abatacept. That was 43 7 out of 8, 73 8 out of 8, 69 received placebo. We did PK studies. We did real-time flow cytometry. We did TCR-seq to understand the T cell repertoire. And what I'm going to show you today as we try to understand why patients get GVHD and how ABA protects from it, we did transcriptomics. This was before the day of single cell transcriptomics. So we sorted CD4 and CD8 cells and did bulk transcriptomics on the sorted populations. I'm going to focus right now for the rest of the talk on the transcriptomics and flow cytometry, focusing on days 21 and 28 before those patients get acute GVHD to answer the why. Why was ABA so successful? I want to introduce you to one of the main tools that we use called weighted gene correlation network analysis. It's a wonderful tool for analyzing complex transcriptional data from human and other complex data sets. This is a computational method used to perform large-scale organization of gene co-expression networks. It permits the construction of what we call a gene correlation matrix from an entire data set. So we added in all of the clinical metadata as well as the transcriptomics and could then link different transcriptomic signatures to different key clinical metadata. And it uses a hierarchical clustering algorithm of the matrix to identify sets of genes that meet a threshold of co-expression. And for ABA2, when we did this clustering, what it identified is a set of 30 discrete self-assembling modules that we see color-coded here on the X and Y axes. And then what we can identify through WGCNA is what is called a module eigengene, which is clusters that also correlate with clinical outcomes. And I'm going to show you some of these in the coming slides. So if you look at these eigengenes here, you can see that there is clusters of the CD8 transcriptome with GVHD and then with the placebo here on the upper left of the WGCNA, very far away from the ABA eigengene. So even just in the WGCNA, you can start to see that there's quite different transcriptome between patients getting ABA and patients not. And very interestingly, that the placebo is really clustering very close with GVHD. It suggests that the transcriptome of these sorted CD4 and CD8 cells are going to tell us something about the mechanism of why abatacept was successful. And indeed, I'm going to go back to the WGC in a second, but when we do differential gene expression analysis, which many people are more familiar with, and we look at what were the genes that were upregulated in the placebo patients that got GVHD versus even just the ABA patients that even got GVHD, what we can see is that they're very different. And in fact, several canonical T cell activation markers are upregulated in those placebo patients, including CCR3, CCR8, TOX, CTLA4. And what's really striking is there were tons of proliferation markers that were upregulated in the placebo patients and really controlled with the CD28 blockade in abatacept. So all of these genes and more that we found are relevant for proliferation were upregulated in the placebo patients. Now, this presents a paradox because I've talked to you about the safety of abatacept, and I've shown you that the T cells reconstituted fine in abatacept. So if abatacept is controlling that proliferation, wouldn't that have led to a deficiency in T cell reconstitution? The paradox is actually resolved if we compare not to patients on transplant, but rather to healthy controls. If you squint down here, you can see that the amount of T cell proliferation in healthy control CD4 and CD8 cells is present, but is very low. And indeed, if we compare both the ABA patients and the placebo patients to healthy control, what you can see is that ABA patients actually, while they did not have the more exuberant and potentially pathogenic, I'll show you that in a second, T cell proliferation that we find in placebos, they did have significantly more proliferation than in healthy controls. And in fact, here is a gene set enrichment analysis, which shows enrichment of T cell proliferation in abatacept when we compare not to placebo patients, but to healthy controls, consistent with this immune reconstitution, the T cell reconstitution that is preserved with abatacept. Now, placebo patients also increase their proliferation compared to healthy controls, but we think there's something more sort of sinister lying beneath that proliferation in the placebo patients. Because when we compare the placebo to ABA, we can see this increased proliferation in placebo versus ABA. But what's even more striking is when we divide those placebo patients out between those that got GVHD and those that did not, those that got GVHD had even more proliferation than those didn't, suggesting that there was some pathogenic T cell proliferation in the placebo patients that developed GVHD. And indeed, if we go back to that WGCNA and we ask, what is the module that is closest to the placebo patients and to GVHD, we see the magenta module, and this module is highly enriched in proliferation pathways, suggesting that proliferation is linked both to placebo and to GVHD. By studying the abatacept patients, we were able to get detailed evidence then that by blocking CD28 and CD8086 signaling, what abatacept does was critical to control that alloproliferation that we saw in the placebo GVHD patients. Let me show you the data that supports that. So here we go back to our flow cytometry, our placebo versus abatacept patient. You can see that ABA patients had less overall proliferation than placebo patients. But let's divide those abatacept patients now out between those that got GVHD and who didn't. So here are the patients that got between 0 and 1 GVHD. But what's really strikingly different than with the placebo patients is if you look at the abatacept patients that develop acute GVHD, you can see that early after transplant, they do show that sort of increase in proliferation. That's after just one dose of abatacept. But from day 5 on, you can see that ABA quenches that alloproliferation, suggesting that CD28 blockade was sufficient to block the pathogenic proliferation that causes GVHD in patients treated with placebo. And again, you can see how different the pattern is between the ABA and the placebo patients. this is underscored by, again, doing another transcriptomic analysis called gene set enrichment analysis. Here, we're contrasting grade 2 to 4 GVHD versus grade 0-1 GVHD in either placebo patients or ABA. And again, you can see that cell cycle or T cell proliferation really marks the patients that got GVHD in placebo, but fails to mark those in ABA. So by baking in these mechanistic studies, we're able to identify proliferation or alloproliferation as key to the pathogenesis in placebo. What happened in those fewer ABA patients that got GVHD? Why did they get it? We were able to look at that as well. Again, we come back to the WGCNA and we ask, well, if proliferation in the magenta module is what really associates with GVHD and placebo, what associated with GVHD and ABA? We find the brown modules and the green modules are most closely associated with ABA. And very interestingly, let's look at the brown module first. Rather than a proliferation module, this is a naive T cell module. It suggests that in abatacept, when you get breakthrough GVHD, it may not be by proliferating T cells, but by naive T cells. And in fact, while this was a surprise and we didn't expect this, we didn't look at this up front in the flow cytometry. When we go back to the flow cytometry for the ABA patients and divide again all naives, and this is CD4 cells, which is really where the signal was most strong, all CD4 cells into those that develop GVHD and not, you can see that there's a significant increase in the naive T cells by flow cytometry in ABA. And again, when we use gene set enrichment analysis, here's where we see enrichment for naive cells in ABA rather than proliferation. The green module also correlated strongly with abatacept. And this was very interesting too, because this is a module that's really focused on translation. And so if you combine the naive T cells with the focus on translation, it really harkened to me a paper that came out in the last year or two, where it suggests that what really primes naive T cells to become activated is that their translational machinery is poised to be turned on, suggesting that in those ABA patients, while we've controlled proliferation, there may be some T cells that still are able to get activated. These are naive T cells. These actually may be T cells that are not only primed to cause acute GVHD, but may be primed to cause chronic GVHD later. So to wrap up what we found with the ABA analysis, what we find is that GVHD is identified by a naive T cell signature. And we've talked a little bit about the cytokine pathway that's important for T cell activation too. And I don't have time to go into the details here, but we do have some evidence that in ABA patients that develop GVHD, there's also increased cytokine secretion. Let me just show you one example here. IL-6 seems to be secreted in patients that have been given ABA, but nonetheless get GVHD. So putting everything together here, let's put together the clinical outcomes, the flow cytometry, and the transcriptomics, and put together a mechanistic model for what causes GVHD in the absence of ABA and how abatacept or costimulation blockade can control that. So again, let's turn back to the model of T cell activation. Remember, we have signal 1 through antigen presentation, signal 2 through co-stimulation, and signal 3, which is the cytokine signal. Let's blow up that synapse between the T cell and the APC. And again, look mechanistically at signal 1, signal 2, and signal 3, and put together a progressive model of control as we add abatacept to CNI and methotrexate. So first, as we're building this model, remember that after transplant, you have ubiquitous antigen around in the patient for the T cells of the donor to see. So this antigen is everywhere. That means that in the absence of immunosuppression, like what we saw in the non-human primate model, you have full activation of signals 1, 2, and 3, and you get massive T cell proliferation, activation, effector maturation. And this can give in the clinical setting, this is actually seen now with another T cell prevention GVHD prophylaxis regimen that doesn't give any immunoprophylaxis for a few days post-transplant. What you can see is actually hyperacute GVHD, or what we call engraftment syndrome, which is really due to this massive T cell proliferation. Now, in our placebo patients that got CNI and methotrexate, what we find is that these partially control signal 1, but really very likely leave signal 2 and signal 3 intact. And what we found here in our ABA2 patients on the placebo arm was that we got classic breakthrough T cell proliferation. We've shown this now by flow, by transcriptomics. And In that proliferation, we get activation, effector maturation, and this is very likely what drives the GVHD, that breakthrough GVHD that we see when patients are only given calcineurin and methotrexate for GVHD prophylaxis. But when you add co-stimulation blockade that can block the CD28, CD80, 86 signal, our data suggests that this is sufficient to lead to a whole different milieu of T cells that successfully reconstitute after transplant. Here, you get significantly improved control of that breakthrough proliferation and effector maturation. And when we do get GVHD, it's breakthrough with these naive cells. And this is really interesting because, as I showed you, one of the really key outcomes of the ABBA-2 study was that ABBA was able to control the steroid refractory GVHD. And we do believe, based on preclinical studies, that naive T cells are often more steroid sensitive, and this may have been one of the mechanistic links with this clinical observation. So to conclude, what I've shown you today is that abatacept improves acute GVHD and survival outcomes in both 8 out of 8 and 7 out of 8 unrelated donor transplants, that the additional immunosuppression with abatacept was not associated with increased relapse or defects in hematologic reconstitution. And then we've gone on to measure our mechanisms of success. We have robust data supporting intact T cell reconstitution in patients treated with abatacept. With both flow cytometry and transcriptomics, we've been able to demonstrate that breakthrough alloproliferation, this pathogenic proliferation, likely drives acute GVHD when you just give CNI and methotrexate, and that by blocking CD28 and CD80-86 co-stimulation, you can control this proliferation. We've also found the signature of breakthrough acute GVHD in abatacept patients, which we believe includes the emergence of a larger naive T cell reservoir in these patients, and with IL-6 signaling very evident in these T cells as well. I think that these data provide a mechanistic explanation for the ability of abatacept to control acute GVHD and nominate new pathways of T cell activation that occur in some patients despite co-stimulation blockade. These are the pathways that we might want to focus on in the future to fully control GVHD in these patients. So to conclude, I'd like to first thank the patients and the families who contributed to the ABBA-2 study, the ABBA-2 study team, my lab at Boston Children's and Dana-Farber, and thank you all for listening.